People, we have to have our cameras on. I have to have some part of your head or your hands or something to know that you're there in the room. Layla. Guys, I don't want to have to stop class to call people's homes or anything like that. That just seems a little wasteful. We have a lot to do today. We have a final next week. Hopefully we'll all be back we'll hopefully all be back in the classroom. But right now, we really need to get people, we need to get to a point where we're doing what we're supposed to.
So like, it sounds like I'm lying, but my camera actually will not turn on.
It sounds like you're lying.
I know, but it actually won't turn on. Like it says like.
I didn't say it sounds like you're lying. You said it
Oh.
sounds like you're lying. I think that's a funny thing to say.
Because it does. It sounds like I'm lying to your face, but I'm not, I swear.
All right. We just have to hurry up and get going because I don't want to waste any time. So come on.
You see it? My camera is on at the bottom.
I see it.
Oh, you want cameras on?
Jasmine, are you here? I can't tell.
Yes, I'm here.
I'm going to use your review. All right. As an example.
Okay, that's fine.
Is that okay? All right. Do you know why I'm using your review as an example?
No, not really.
You can't guess why?
Nope.
Well, you did an excellent job, first of all, and you went above and beyond. You took really good notes and you did every part of it. So I think that's really excellent. Okay. And I just want to use it as an example for what we're doing here.
Okay.
I wish I could, I wonder, if I guess I have to download, I'm gonna have to download every section separately. All right. So I gotta say, some of you haven't turned in that review, which is your prerogative. I mean, obviously it's gonna be due at the end of the week. You won't get any points for it. The final will be next week, right? Remember, we're being recorded. I should remind you of that, even though I think it says on your screen, doesn't it? Doesn't say it's recording on your screen?
Yeah.
Yeah, I was hoping it would say that. So anyways, you have, trying to get this straight here. I think I only have one or two people that have actually turned in the review that opportunity you had to do over break. Only a few of you actually did it, and it's unfortunate that you didn't take advantage of that opportunity. Let me see.
I wasn't here the last week, so I didn't know if we had a review.
I'm sorry to hear that. I did post it. It is posted on Schoology. Did you see that?
No, I don't remember seeing it.
Okay, well, it's there. So try to, you'll have to just get it done.
Mr. Mendoza, I wasn't here for the last week's of school really either.
Yeah, darling, everything's posted on Schoology, right? Even that's why I've been telling you guys forever. You guys are in class. If you miss class, everything's posted on Schoology. I don't know how else to say it makes any kind of sense. Does that make sense to everybody or not? Does anybody have a problem with getting into Schoology, for instance? I'm asking a question. Could I get some answers? Fudge, Jemiah, Kimberly. We have to get our cameras on. All right. Hello. Oh, wow. It's going to take half an hour to get you guys just even to. Hello. Wow. Okay.
You breaking up.
I need you to, Kimberly, Jemiah, to turn your cameras on. And I need to see some part of your head or hands so that I know that you're actually in the room. That is a requirement for me. Now, I don't know how else to say that except to say it that way. So let's get it done. Turn the camera on, please. I understand you might need to get your hair done or whatever, or quickly put something on it. I don't know, whatever the issue is, but I need to see that you're not in bed asleep. You're not in the room. That's not acceptable. So I need to see that. And those of you that did not get a chance, that were absent the last week of class because you had contact with someone who had COVID, or you had COVID, or you went on vacation, or whatever the situation was, everybody had a different reason. That's fine. That's not a problem. The work is all there. Next week, the grades are going in. The grades are going to be what they are. And if the work's not turned in, it's going to be a 0, right? So you gotta get that work turned in. I mean, that's all there is to it. Now, those of you again that missed, that's fine that you missed. We're going to go through the review this week, but you have a big final next week. And that final is going to be a big part of your grade. So I really feel strongly that you guys need to make sure that you know what you're doing. And we're going to go through that review this week, but the work is also due, and the work is worth a lot of points. Is that clear to everybody? Have I made myself clear? Yes or no, please?
Yeah.
All right. So I'm going to go ahead and we're going to look at this, at Jasmine's work. She turned in 70 pages of work. I don't know why that took so long. She turned in 70 pages of work that she did over break. I'm assuming, Jasmine, did you do it over?
Yeah, I did it over break.
All right. So Jasmine did it over break. And how hard was it for you to do over break, dear?
I think because of the way I did it, it wasn't that bad.
Okay, how did you do it? Just to share with other people. I just want to emphasize with other.
I kind of just did it like day by day in pieces.
Okay, yeah. So you did day by day in pieces, and it wasn't that hard. Okay. Other people that are going to try to get all this work done over this week, we're going to do as much as we can in class. How long is this class going to last today? Do you all know? When is this class over?
30 minutes.
30 minutes. And so we're not going to be able to waste time like we are today, like asking Naya Crawford to go ahead and turn her camera on, and Jemiah and Jada to turn their cameras on, and make sure that I can't waste that kind of time from now on. We only have 4 days. So if you don't turn your camera on and you don't give me a reason why, you don't come and talk to me why, I'm gonna have to call your folks. We're gonna have to figure out why it is you can't get your camera to turn on. I know I have one person who has a technical issue, I believe her. But if you can't participate, then we're gonna have to have a discussion, right? Because I'm gonna have to make sure your parents know that if you don't pass, you know, if you're not gonna pass the class, why is it that that's happening? They can't just be surprised. We're not gonna hide that from your folks, right? So again, Faith, Jemiah, you're signed in, Jemiah, 3 times. Why? It's really strange. It's okay, but are you okay? Are you there? Did you just all 3 log off? I don't understand what's going on. Can someone text her or something and let me know why, what kind of issues she's having? They seem very strange to me. All right, well, let's look at on the screen what we have, where this is the review that we were going, that Jasmine did. Jasmine did an excellent job. She got all the 100% because the whole objective was to get it done, not to get it necessarily right. I'm sure she has most of it right. I'm just saying it's not, that's being right was not the objective. So let's take a look at the first question. First question was, how are organic molecules related to all living things? They make up all living things. That makes sense, right? You should be taking notes, people. You guys should be taking notes.
I can't see it.
It's on the screen. You can't see the screen? Can everyone see the screen or not?
Yeah, no, it was just lopsided.
Oh. But the screen is visible, yes?
Yeah.
Let me put the chat up. I see some people are chatting. Oh, it's okay, Kim. Yeah, whatever you gotta do. Yeah, you don't have to be on all the time. If you gotta leave for whatever reason, just put it in the chat. That's fine. You can put it directly to me if it's private. You don't have to share it with everyone. You all know how to do that, right? How to send a message directly to me rather than sending it to the whole class. Does everybody know how to do that? No? Yes? Maybe?
Yes.
Okay.
Yeah.
All right, so let's go ahead and take a look at this. So the organic molecules make up most or all make up living things. That's what we're made of. That's what all living things are made of on this planet, organic molecules. What is organic molecule? What are organic molecules? And at this point, I hope you know that, yes, they're carbohydrates, right? Is that correct? Yes? Could you guys, I'm not watching the screen, so I need you either to unmute and say something or to go ahead and put in the chat if you'd like, but I need to see it, or a thumbs up is great, but I won't see it. Karina, if you could keep an eye on the screen, if you see a thumbs up, let me know, because some of these kids are a little shy on talking.
Everyone has the thumbs up on.
Thank you. So carbohydrates, proteins, lipids, and nucleic acids, we know that those are the 4 main biomolecules. We talked about them. We called them biomolecules or organic molecules. Organic means that they have carbon. That's what organic means, right? Molecules that have carbon in them. So we know that all these molecules have carbon in them. We've drawn them. We know what they look like. You should. I'll be clear. If you don't know what they look like, it's going to hurt you on the final. By the way, both parts of the final are already listed in Schoology, right?
Yes.
Sorry, what? Oh, okay. So all organic molecules are organelles. No, they're not organelles. Organic molecules are molecules that are made of carbon. The 4 types are carbohydrates, lipids, proteins, and nucleic acids. Now, if you put a bunch of these organic molecules together, you will have organelles. That's correct. The organic molecules make up organelles. Now, at this point, if you don't know that, it's just going to take you a little more effort to get to where you need to be for next week. And then organelles make up what? A bunch of organelles together make up what?
Cell.
Cells. That's correct. So they make up cells. All right. So we've talked about the organelles and how they work. We talked about carbohydrates, proteins, lipids, nucleic acids. We've drawn them. You should be able to do a quick sketch. So with carbohydrates, you should be able to draw something, if you can draw looks like this, a glucose molecule or a pentose molecule, remembering that one of these is oxygen. Then that's that this is these are carbohydrates in ring form. And we know at this point that, or at least you should know, that they're carbon, hydrogen, and oxygen are what the elements they're made of, and that they're 1 to 2 to 1 ratio. So at this point, carbohydrates, you should have this information in your head. If you don't have this information in your head, boy, you need to start putting it in there before next week, because it will be tested. You will have to identify them on your FRQ, on your FRQ day, on your multiple choice day. You'll have to identify some of them. You'll have to tell the difference between a carbohydrate and, let's say, a nucleic acid. Let's look at a nucleic acid. A nucleic acid is made of what? Who can tell me what nucleic acid is made of? What elements? What's one element everyone knows nucleic acids are made of?
Proteins.
Nope, that's not an element. That's a biomolecule. So I need to correct that.
Carbon.
Carbon, yeah, pure carbon, hydrogen, nitrogen, phosphorus, right? Oxygen. So nucleic acids had all these elements in them. And in nucleic acids, in humans and most nucleic acids on this planet, for most living things on this planet, what we're dealing with is a ribose or deoxyribose, right? There's two. It's either ribose, nucleic acid, which stands for what? What do we call ribonucleic acid? If something's a ribonucleic acid, what do we call it?
RNA.
RNA. Or it's a deoxyribonucleic acid, which is what?
DNA.
That's it. That's all there is to it. So what's the difference? What's the difference between the two? Let's take a quick look. Obviously, you should know more. Again, looking at the review sheets, looking at your notes. If you're sitting there quietly confused, then that's something that you really need to to work on, because you literally have run out of time. So we're looking at oxygen. You have a 5 carbon ring structure. Now, ribose are 5 carbon sugars. There's the first, there's the second, there's the third, there's the fourth, and there's the fifth. There's 5 carbons with a little prime on them. So these are all 1 prime to 5 prime, right? And the difference between RNA is RNA has this oxygen sitting here on the second carbon. So there's an oxygen here sticking off. There's an OH group here. And that's why we call this ribonucleic acid. Deoxyribonucleic acid, what do you think about this one? What's the difference between these two ribose sugars? What's the difference between deoxy and and ribonucleic acid, RNA and DNA. What's one difference? All these are sugar, all these are carbons, what I'm labeling now, 1', 2', 3', 4', 5'. These are all carbons. These are oxygen. These two are the same. So far, the structures are the same. Do you agree?
Yeah.
Yeah, they are the same. The difference between deoxyribonucleic acid and ribonucleic acid is what? What is the difference between the two?
One
Doesn't
has an
RNA have
OH and one
C?
has an H.
Good, I'm glad, and we're getting people motivated. I love it. We'll say that again. Go ahead and say it.
One has an OH and one has an H.
That's right. Excellent. That's it. That's it. Yeah. This deoxy doesn't have the oxygen, only has the hydrogen on that second carbon. Does that make sense to everybody? Deoxy ribonucleic acid versus ribonucleic acid. You could get a question like that on a test. There could be two different nucleic acids, and you'd be asked which one of these is DNA and which is RNA. The other common thing that they both have is that they both have phosphate, a phosphate group sticking on the 5 attached to the 5th carbon. Is that clear?
Yeah.
Okay, so those are the kind of the main things. You got two ribose or two 5-carbon ring structures that look like home base, right? The pentagon. The RNA has an OH group on the second carbon. The DNA has only hydrogen on the second carbon. They both have phosphate on the 5', so that's the same. Then you have a third similarity, and that's that on the first carbon, off of the first carbon, you have a nitrogenous base. It's either a single ring or a double ring nitrogenous base. What are the nitrogenous bases for RNA? What are these parts here that stick off the side of ribose? What are they for RNA? Does anybody know? You should know. Let me put it that way, in case you're forgetting.
Is it like glucose? Something like that?
Glucose, no. Only ribose. Glucose is 6-carbon sugar. It's a carbohydrate. This is a nitrogenous base, which suggests it has what element in it, by the way?
Nitrogen.
Nitrogen, correct, yeah. Jacob, Tyler, Jemiah, Jada, please try to turn your cameras on. I don't want to have to spend my time 4th period calling parents. Just need to see this. You're there in the room. Unless you're, if you have to leave for whatever reason, send me a direct message. All right. Nitrogenous base. Well, you can have A in RNA, or you can have what?
T.
You can't have T in RNA, but good guess. T, you can have
U.
You can have U
C.
E.
and G. And in DNA, you can have A, T, G, and C. So that's the difference between these letters, is which nitrogenous base is sticking off the side here. And we've looked at the details of them. We looked at their structure. No, you don't have to know the difference, how they look, what they look like. Is that clear? You do have to know the ribose. You do have to know that there's an OH group here sticking off the second carbon. You need to know the phosphates here. And by the way, let me remind you that phosphate is, that phosphate that you see me drawing a P with an O is actually phosphorus with 4 oxygens. That's what this P stands for, okay? I don't want you to think it's just phosphorus. So that's DNA and RNA. We know, by the way, that when you connect these together, when you take this ribose sugar and you with its phosphate, and it's nitrogenous base sticking off the side, whatever that is, the double ring, whatever it is, whether it's RNA or DNA, right? No matter what, whether it's RNA or DNA, what we do see is that we can connect these. And so this is the monomer. This is the monomer of DNA and RNA. So we can take these and we can connect them in a chain. As we've talked about, we can make a polymer of these. And the way we do it is we attach, or the way life does it is it goes from the 5', remember this is a 5', you can look at the numbers that we drew a minute ago. This is 5' here, from 5', from 3' to 5', we can connect them like this and put them all in a chain. And they start to twist because of their shapes, because they push away from each other and the geometry of them. There's a shape that is forced out. Now in RNA, if you have RNA, it's just it can be many shapes. So RNA can have many different shapes. It's a single chain, a single strand. There's no double strand. Is that clear? It can have all kinds of shapes. You know that. You know that, for instance, our RNA is ribosomal RNA, and it takes the shape of a ribosome. Looks like that. There's various forms and twists and turns, but they take shapes like that. You know, tRNA, the transfer RNA, actually looks like a T as far as I'm concerned. I think it looks like a T anyways. And you have all kinds of other RNA. You have messenger RNA. It's just a single, it's a strand that has a poly A tail. You know this all, right? We went over it. It has a 3' cap. There's a poly A tail, and then there's a message in between. So they have different shapes. RNA can have different shapes. Interestingly, DNA doesn't. What does DNA have? What shape does DNA have? First, is DNA double-stranded or single-stranded?
It's double-stranded.
Double-stranded, good.
Yes, ma'am.
Double-stranded is correct. So then it's double-stranded. So you have one strand, and then you have the second strand. And we call that double-stranded, and it's a double helix. That's why we call it a double helix. It's a double alpha helix. That's the Greek letter alpha. The nitrogenous bases, they hold a bond in between the double strands, holding them together. So when you're looking at the nitrogenous bases, the two strands are held together by those things called hydrogen bonds. So hydrogen bonds stick the two strands together. The hydrogen bonds happen between the A and the T, ATGC, right? That's the matching stuff. That's where they match. So remember, anytime you have an A on one strand, what do you have on the other?
A T.
In DNA, the T matches the other. So this is a hydrogen bond. That's what holds the DNA together. And then the G is matched with the C. What about RNA? How do they, if it does do when it does these shapes, when it twists and turns, when it does its matching, how does it match?
A to U.
A to U and then G to C. And it has to do with geometry and some molecular chemistry, but the bottom line is that's how they bond. That's how they attract each other that's how they make shapes in nature. They attract each other like magnets, right? These aren't sharing electrons. They're not a strong bond, but even though they're weak intermolecular force, they do define shapes. So that's carbohydrates, that's nucleic acids. One other thing you should know about carbohydrates is that it has a monomer. There's several different kinds of monomers. You know, someone mentioned a moment ago, glucose, that's a monomer. You can put them together, you can put glucose together in a lot of different polymers. So this is the monomer. Just like nucleic acids, you can put this glucose together in a chain. You put it in one chain and you get something called glycogen. That's what you put away in your liver. It's a bunch of glucose molecules all put away in a chain. The difference between glycogen and, let's say, another one, that wood that you sit on your stools at school, that's another kind of polymer. These are polymers.
Mendoza, class is over.
Oh, okay. Well, you have a good day, and try to make sure that you get to class and be here tomorrow on time.
Mr. Mendoza,
Yeah, somebody has a question.
I changed my name. It's a Mark.
Mendoza.
Mark. I see Mark High. Yeah, Mark Verify.
I can't hear you. Say that again.
Mr. Mendoza, can you hear me now?
Yeah, go ahead.
So students have said they got like a packet of winter break, and I wasn't at school.
They did, yeah. And it's posted in Schoology. That's right.
Okay, thank you. And is this video gonna be posted later on?
Yeah, yeah. I'm recording. It's being recorded now.
Okay, thank you.
Well,
Mr. Mendoza, is all the videos from like the beginning of the school year until now, is that all on your YouTube page so I could take more notes?
they are. I would focus on maybe coming and seeing me and asking questions, but all the classes that are posted, they're all in Schoology. Most of them
Okay.
is in my on the new class YouTube channel. Yeah.
Okay.
Let's see. All right. Have a good one. All right. So I need someone to be a co-host. Who wants to be the co-host? Jada Gardner's still here, which makes me think I don't know why. Jada's still here. I'm wondering if she's even. All right, let me just go ahead. Jada, you there? Jada.
Yes.
Okay. No, that's Covington. Jada Gardner. Jada Gardner. All right, I'm going to go ahead and put you out, Jada. Well, I don't want to do that because last time I put someone out, they weren't able to come in, but I better leave that alone, huh? You're right. All right, who wants to be the co-host?
What do you mean by that?
All right, Jasmine, you're co-host. Thanks for volunteering.
Oh no. Okay.
All right, so Jada, what I need you to do is I need you to, anytime somebody asks to come in, I need you to admit them. All right? And we'll try to make a different person to co-host all week. Does that make sense? So go ahead and let Charlize in now. And Abriana. All right. If you have I need everyone to turn their cameras on, and I need to see some part of either your hands or the top of your head at least. I don't care. I don't need to see your whole face, but I need to see that you're in the room. So throughout the lesson, so sticking your hand out, pointing the camera to the ceiling, and sticking your hand out every once in a while is that really going to work? I'm not sure. but you had, oh, I understand your camera's probably back of your computer, so that sucks. But okay, I get your point. So just need to see that you're in the room. I've had already today, I've had a few people who I'm wondering if they were even in the room at all. So I'll have to call their parents and have a discussion with them. But I'd rather not have to call your parents and have a discussion whether you're actually attending class or not. So I just like to make sure I see some part, either your head or your hands. If I don't see that, I'm gonna have to ask you why, and I'm gonna have to have that conversation with your parents. So I that's all I can say about that. So Iwais, Jaya, Josiah, and everyone else, please make sure your cameras are on and that I can see some part of what you're doing. Could someone let Rivera in? I guess I'll admit him. All right. So you had an assignment. You haven't been able to grade those reviews yet. They were due today. Jasmine from the other class allowed me to use her work. I'm sure a lot of you have done your work already. Jasmine's a ninth grader, but she's done a fantastic job. And she's allowed me to use her work. She finished all 70 pages of the review. And when I say 70s, because she took a lot of notes as she was doing it. She actually wrote a lot of interesting notes. I feel like I should let you see what she's done before I continue with the review. I know some of you were absent during that last week or two of class. That being said, yes, that's fine. You were absent. There was a handout that I handed out. Some people got it, some people didn't. That's also fine. It's posted electronically. It's all posted electronically on Schoology. The YouTube videos for the class lessons are all posted on Schoology or on the YouTube channel for the class YouTube channel. So there should be no real reason why people don't have access to everything you need. There's absolutely everything that you think that's going to be on a test is there, has been made clear to you now for something like, I think, almost a month now you've had this work. Next week is the final. It's the end of the semester. That's it. It's the grade that you're going to get. Some of you have really good grades now. If you mess up the final next week, it could bring your grade down. Yes. I know that that's a question some people have. By how much, it depends. It all depends on various factors. We can have a personal conference if a grade conference if you really feel like you need to. You know when the office hours are. I'm going to be available for you all week long. If you're missing work, I would turn it in. If I were you, I would make sure all my missing work is turned in. I would do this review that you've had for about 3 weeks now. I would make sure I do it if I were you. I would not waste any more time. I know some of you were busy. Some of you took a break. You had that choice. It was only extra credit over the break. It's not a big deal. Jasmine shared with us in class that it really wasn't that hard because she only did a few pages a day and she was fine with it. But if you really needed a break, that's fine. You're back in school now. It needs to get done by next week. So this is the first page of the biomolecule. I'm going to go ahead and just flip through it on the screen. Do you all see the screen? Do you see the screen? Yeah. Okay.
Yeah.
Guys, I need you guys. I'm not going to be looking at the screen all the time, so you're going to have to speak. Or someone's going to have to speak. If somebody puts a thumbs up, somebody's got to say, hey, people are putting thumbs up or something. All right. I can't be looking at the screen and giving you notes at the same time. Everybody should be writing notes. Diaz, your camera's off. Simpson, your camera's off. Abriana, Jaya, Nolanix, your cameras are off. One more time. All right. So here's here's her page. I'm just going to flip through it and then we'll go back and start the review that we're going to do all week long. All right. So these are the questions she's answered on, those were biomolecules, right? And then here's transport across membrane. And then here is some stuff on more osmosis practice answers. Here's her notes, all the vocabulary words she's put together, color coordinated in charts. In fact, I almost feel like I should send you all this because it's just such great work. Then she went on and continued and completed the cell preparation questions. Again, these are practice questions. She did all the practice questions. It has all the vocabulary words. These are kind of simplistic, but she got, as far as I can tell, all of them right. I haven't been able to grade them in detail. All the organelles, what they do, bacteria versus viruses, diffusion and osmosis questions. These are all practice questions that she did. And all the answers were there at the end of the unit anyways, as well as the glossary. And I think you noticed that she took notes and rewrote all her definitions herself. This was not asked of her, but she did it anyways. Here it is again. And yet one more, the words that she did not define, she redefined for herself again. And then here is her transcription translation practice stuff. And she completed this. She did it digitally, but she made her RNA and DNA. And I think I put the notes in twice, or that was my mistake. So it's not 70, it's more like 60, I guess. So anyways, that's the work she had to do. And she said it didn't take her that long to do it. I think it's excellent work. And we're going to go ahead and start to go over this material now. I expect you guys to take notes. Don't be surprised if I ask you to upload the notes at some point. Simpson, Jaya, is there a reason why your cameras are not on? Simpson? Diaz, why is your screen black? Are you guys able to speak? Can you put something in the chat? All right, not sure why your camera's black. I'm sure, I'm assuming, but you are at least there because you're responding in chat. Okay. Kylie, your camera is on, it's pointed to the ceiling, but the fact that you're participating in the chat lets me know you're actually there and listening, which is the key here. I won't be looking at the screen. I made someone the co-host, right? Why is not co-host not allowed? All right,
Yeah.
All right, so when you see someone in the waiting room, you gotta let them in. Is that okay?
Yeah.
All right. All right, so let's go on then. So the first question is organic molecules, and organic molecules are obviously, at least I hope they're obvious, they're things that are made of carbon. They make up living things, and they're made of carbon. Everybody understands that, yes? Organic molecules that are made of carbon then come together and form organelles, and organelles come together and make cells. That should be clear to everybody. If it's not, then you have some work to do. You have carbohydrates, proteins, lipids, nucleic acids. So there's four main kinds of organic molecules. Another word we use is biomolecules, the molecules of life. Organic just means they're made of carbon. And you know by now that most of these elements, most of these molecules are made of carbon. This here's an example of a monomer called fructose. So there's the oxygen. Now let's count this. Let's count these fructose. So this is one, carbon 1. Here's the second prime carbon, the third prime carbon, the fourth prime carbon. Oops, I messed up. Second prime, third prime, fourth prime, fifth prime, and sixth prime. So there's 6 carbons in fructose, 6. even though it makes a pentose ring. You see it makes a pentagon? Do you see the ring is a pentagon? Yes or no?
Yes.
Okay. You guys have to answer. I can't be looking at the screen and write on the iPad. So this is unusual because usually a 6-carbon, when you think of a 6-carbon ring, you think of 6 sides. So hopefully you're thinking of something like glucose. And glucose and fructose, their big difference is how they're put together. I should make sure I do that. So glucose is a 6-sided ring. So you have 1 prime, 2 prime, 3 prime. Oops, that shouldn't be there. 4 prime, 5 prime, and 6 prime. So it's a 6-sided ring. It has 6 carbons, and that's glucose. When it makes a ring, they're not always rings. Sometimes they're straight chain, but in any case. So carbohydrates are made of carbon, hydrogen, and oxygen in a 1:2:1 ratio. Does everybody remember that? Is that a yes, a no? I don't know. Like, can we put things in the chat? Can we participate? Is that asking too much? Okay. And we don't have time. How long do we have in class?
Like 30 minutes.
Like 30 minutes. All we got is 30 minutes. We only got till 9:05. It's 8:47 already. So we have no time, especially starting tomorrow. We have no time for me to sit here and try to talk to you into turning your camera on. Simpson, are you there? Are you in class? Can you unmute and speak if you can't turn your camera on? Hello? Could someone text her and ask her? Carmen? Could someone text Carmen and ask her what's going on? Well, she seems like she's jammed up, so I'm going to go ahead and put her out of class, and then she's going to have to rejoin, or ask to rejoin, I guess. See, that's the problem, is that I can't remove her without kicking her out permanently from the class. This is really upsetting. I'll just have to call her parents, that's all. Let her know I'm gonna have to have a conversation with her folks to find out what's going on, if it's a technology issue or what have you. If somebody could text her, I'd appreciate that. So we have, when it comes to carbohydrates, they're a 1:2:1 ratio. So you have a 1:2:1 ratio. So when you're looking at something like glucose, it has 6 carbons. How many hydrogens does it have?
12.
12.
12 is correct. That's 12 hydrogens. How many oxygens?
6.
Correct. When you're looking at fructose, which we just looked at a minute ago, it has 6 carbons. How many hydrogens does it have?
12.
12. And how many oxygens?
6.
That's correct. So it has the same number of oxygens, hydrogens, and carbons, but they're just put together differently. And so they make up 2 different molecules, even though they're the same atoms. What about something like the other monomer or carbohydrate that we know of as ribose? What about ribose? It has 5 carbons, not 6. How many hydrogens?
10.
10 is correct. And how many oxygens?
5.
So now ribose is substantially different. It's not only a difference in shape, it's also a difference in number of molecules. So let's take a look at ribonucleic acids, because that's another kind of biomolecule, right? Organic molecule called nucleic acids. And we know that they're made up of carbon, hydrogen, nitrogen, phosphate, and oxygen. So where where carbohydrates are just carbon, hydrogen, and oxygen, nucleic acids have carbon, hydrogen, nitrogen, phosphorus, and oxygen. The big difference, obviously, I hope everybody sees that the big difference here is that nucleic acids have nitrogen and phosphorus. That's the carbon, the glucose, the carbohydrates don't have these two. And again, did someone text Carmen to let her know that to find out what's going on with her? She's okay. Is someone doing that? Is that a yes? No? No? No one? Okay. Oh, well. All right. So there's two kinds of there's ribonucleic acid and deoxyribonucleic acid. Let me ask you, what's the difference between the two? The difference between RNA and DNA. Give me the differences. Everybody should know this by now. You're right. This is something that's expected. There should be no surprises here. What's the difference between ribonucleic acid and deoxyribonucleic acid? The silence worries me. So somebody needs to speak up. It's 8:52. We are up till 9:05.
One has sugar and the other one does not.
Well, that's a good guess. No, they both have sugars. They both have a 5 carbon sugar, right? A ribose. There's a difference between the ribose. Any other guesses between the? If you don't know the difference at this stage, you really have a lot of work to do. Really does worry me somewhat that you don't know the difference. The final is next week, whether you're ready or not. I just hope you get ready. So the difference between, this is something that's very disheartening to hear that you don't know the difference between DNA and RNA, but the difference is that one difference is that the second carbon, second position of the ribose, remembering we start with the oxygen, we number it around 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. As we go around on the second carbon in deoxyribose nucleic acid, you have a hydrogen. In RNA, you have an OH group. You will have both these molecules on the test. And if you need to be able to tell the difference between those major structures. So make sure that you have that difference embedded in your mind. All right. At the 5 prime, both RNA and DNA have a phosphate connected to them. They have a phosphate connected to them at the 5 prime. And at this 1 prime, at the first prime, the first carbon in the ring, there's a nitrogenous base attached. So that's pretty much a nucleotide. That's the monomer of both RNA and DNA. So the big difference between RNA and DNA on the basic structure is the OH, there's an oxygen on the second carbon in deoxy, that should make sense to you, right? No, D means lack of oxygen. Deoxynucleic acid, there's an oxygen missing on the second carbon. So that should be pretty clear by now. What else? What other differences do you know between DNA and RNA? You should know a bunch of differences, at least 2 more major differences. If you don't, you got problems. Another difference between DNA and RNA.
Is it the sequence between them? The T becomes like a U?
That's right. That's right. Excellent. So in DNA and RNA has AUGC, the nitrogenous bases, right? Are A, U, C, or G. One of those can be in there. Now, 2 of these are double ringed and 2 of them are single ringed. That's fine. You don't have to know that. You don't have to know how they look differently. You do not have to know that. You do have to know that the ribose is 5 carbons, that it's a ring. There's an oxygen here. You need to know that there's an OH group on the second carbon. You need to know that there's a phosphate here, a nitrogenous base there. That's all something you should know. Here, you should know that RNA has AUGCG, AUCG, and DNA has ATGC. The difference being, again, to be clear, the difference is that there's a U in RNA where there's a T in DNA. That should be clear with all the work that you've done before break, and certainly clear now that you've had a chance to do the test review. Obviously, some people haven't done the test review, so you just have to work a little harder this week. All right, so when you look at these nucleotides put together, these are monomers, so we can put the monomers together, whether they're DNA or RNA. We put them together in the same way. We go 5' to 3', so these are connected. There's a 3' here, there's a 5' here, there's a 3' here, see a 3', then they just keep going. We call this the phosphate sugar backbone of DNA and RNA. That should make sense to you, yes? Why do we say phosphate sugar backbone? The silence is killing me. What's the difference? Why do we call it the phosphate sugar backbone? Jada Covington, why do we call it the phosphate sugar backbone? I have to actually ask an individual. No, very little participation here. You're killing me. Jada, why is it called a phosphate sugar backbone?
Because they're all attached, and that's the only way they can attach.
That's not a bad answer, but take a look at what you see in front of you and think, why phosphate sugar? Do you see a phosphate there in front of you?
Yeah, and then it attaches to the sugar ribosome.
And then it repeats, doesn't it?
Yes.
So it's phosphate, sugar, phosphate, sugar, phosphate, sugar. And it looks like a spine. Doesn't it look like a spine in an abstract way? Yes or no? Have you seen a spine?
Yes.
Skeleton, does it not look like a bunch of vertebrae in a spine? Yes? People?
Yeah.
Okay. So a spine is, another word for a spine is backbone. And that's why they call it a phosphate sugar backbone. So phosphate sugar repeated. This is called a polymer. It's a polymer. It could be a polymer of DNA. It could be a polymer of RNA. But a polymer it is nonetheless. What is a polymer? A bunch of repeated units. Repeated units and repeated monomers. The monomer here is called the nucleotide. It's repeated over and over and over again. Of course, is it DNA or an RNA? At this point, we wouldn't know because we'd need to know if there's an oxygen here at this question mark. Do you see that? Yes? You'd have to know, is this, if there's an oxygen here, then you know it's RNA. If there's no oxygen there, it's just hydrogen, then you know it's DNA. Did someone just leave? I have to look at that attendance. So when we look at RNA, or rather, the other thing that we have to remember is that AUCG, that ATGC, those are all here. Those are here. That's what this is. A would be here, or T here, or G here, or C here. If it's DNA, or A here, or U here, G, and then C, whatever. Whatever the sequence is, this, not that, the difference between A, U, C, G, T, all those different, that is just the nitrogenous base. That's this piece that sticks out. We're all clear on that? Okay. You should know that by now. I don't, if you don't know that by now, wow, I don't know what's going on. I don't know what's going on. So here's RNA and DNA, some more differences. RNA is double-stranded, has 2 strands, and they're wrapped around each other. the nitrogenous bases, those green little bases, they hydrogen bond to another, they stick to one another. So they form a double helix, a double alpha helix. You should be writing this down if you don't know it, because I promise you it's going to be on the test. And what holds the two strands together is the hydrogen bond between A and T and the hydrogen bond between G and C. A and T sticks together and G and C stick together, and they form this double-stranded bond. And these are known as base pairing. The hydrogen bonds base pair. Or rather, the base pairing means they're hydrogen bonding with one another. The geometry of it means that if there's an A on this side, there has to be a T on this side. And if there's a G on one side, there has to be a C on the other. That's the geometry of it. There's no way to fit it all in the right place. We did a lab where we put them together, did we not? Did we not make a paper lab? We cut it all out. We put it all together. We read the chapter. We did practice problems. We did a lab where we put it together with a three-dimensional model. So so not only did you cut it out and make a three-dimensional model in paper, but you also put it together as a three-dimensional model in class, pre-made three-dimensional model. So I'm trying to stimulate your memory on what you did in class that you should. And the reason why I expect you to know this is because we spent so much time in class doing it. Then RNA, as we discussed, is single-stranded. It's not double-stranded, where DNA always comes in pairs that are wound around each other in alpha helix. RNA is single-stranded, it's just one single strand, but it does wrap around. It does make different shapes. It does stick to itself. Where DNA has two strands that stick to one another, RNA has one strand that sticks to itself, and it can make different shapes. Our RNA can make this thing called the ribosome, right? And tRNA, in my view, looks like a T, but that's up to you. It's called transfer RNA. It carries amino acids in the translation portion of protein synthesis. We know that, right? We all talked about it in class. This is tRNA, and it carries amino acids to the ribosome. And then there's this single-stranded RNA called mRNA that carries the message to the ribosome. So those three portions that's what RNA is. Again, it's just this strand of a bunch of these polymers of nucleotides stuck together with different A, U, C, or G in the strand that makes it have a specific shape. If it's rRNA, it looks like a ribosome, acts as an enzyme. If it's tRNA, it looks like a T and it carries acids. Why are people still coming in so late? Are these people early? I don't even know. mRNA is a long chain that carries a message to the ribosome, and they match up with A matches with U and G with C. Are we all good with that? All right, that's it. A yes, no, maybe?
Yes.
Yeah.
Okay. And remember that glucose or carbohydrates also form polymers, things like glycogen and cellulose, but we're going to have to wait on that till tomorrow because I think our time is up here. So glucose can also be connected in chains, different shapes, and depending on the shape can form wood, those chairs you sit on in class, or it can form glycogen in your liver, either way. All right, well, we'll talk more about glucose tomorrow. Remember, we only have half an hour, so join in quickly and get to your next class now. Have a good one.
You too. Thank you.
Bye. All right, who wants to be the co-host? Oh, thanks for volunteering, Sierra.
Wait, what? What I'm doing?
Everybody turn your cameras on, please, and thank you. And so you are now the co-host, Sierra. Anytime someone's in the waiting room, I need you to let them in. Okay, they're gonna be doing that throughout the period. There'll be people coming in like a minute class is over for some reason, but just admit them. Can you do that, Sarah?
Yeah.
Thank you. We all need to have our cameras on. Anybody who doesn't have the camera on, I'm gonna have to have a conversation to figure out if you're actually in class or not. I need you to participate in class. We have 1 week and then we have our final. I mean, this is it, guys. It's your grade, whether you redo biology again this semester or not is up to you. Some part of your room, I need to know you're actually in the room. We can turn on our cameras, it'd be great.
Oh, you suck.
Oh, I know it. I'm the worst.
You're the worst.
Who's speaking? The voice is coming in like 8 minutes late.
Bro, nigga.
It's Trenton, that's who it is. All right.
Record. Oh, come on, we gotta highlight my phone. I can't remember.
Turn your cameras on, guys. You can hide your faces. I don't care. Put your hoodies on. I just need to know that you're actually in the room. That's all I need to know. See the top of your head, your hands. I don't care, but I need to see that you're in the room, not just the ceiling, because I can't tell if you're actually there or not. Thank you all. Where's my co-host? Sierra, let them in.
They're journey.
They're annoying. Are they not annoying when people come in late? Welcome to my world. Bing, 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 bing. Maya, Amar, Christian, both Graves and Kate Christian. Sana, Janaea Reed. Let's have the cameras on, guys. Serenity. I wonder why can't I get everybody in? Class is over at 9:39. We only have 30 minutes left. I'd rather not waste our time. Christian, Willie. I don't understand why people can't follow directions. Serenity, Maya. Maya, Colbert.
Yes, Mr. Mendoza.
I turned the camera on, man.
Why?
I need to know you're in the room, man.
You can hear me
Are you going to talk all throughout the entire period?
if you want me to.
I need to hear you're there. And those of you that have your cameras up to the ceiling, I need to hear that you're actually in the room. That's a cute little fireplace you got there, whoever that is. Who is that? Jay, I love your fireplace.
Hello.
All right. So let's go on. We have a final next week. We only have 5 days this week. Class is over at 9:30. So we only have half an hour a day. I don't know what to say to you except that if we're not, let me put an alarm for 9:29. if you're not prepared for the test, it's gonna be hard for you to do well in the class, especially if you don't have a great grade right now. The final is a big part of your grade. It's not the only thing. You had your review. This is the review. This is a ninth grader actually volunteered to let me use her work that she did over break. What she shared with us in class was that it was really easy for her to do because it was just a little bit every day. But those of you that didn't do it, you have till the end of the week to get it done. It's a review for the final, so the final it'll be due next week if you didn't turn it in already. And again, it was voluntary whether you did it over break or not. You've had it for about 3 weeks now, 4 weeks, almost a month. You have your final next week. It covers everything we covered in class, all the labs, all the activities, all the material that you're expected to be able to do. So there really should not be any question on what is on the test and what you're going to be and what you're being assessed on. So let's go ahead and start. First question on, oh, by the way, I should let you look at the work she's done. It says 70 pages only because I unfortunately made put material in twice. I had to airdrop it from my laptop. But you can see the questions she answered over break. Then there were biomolecule questions. What's an amino acid, RNA, DNA? Then there's a transport across the membrane, which osmosis diffusion, endocytosis, facilitated diffusion, exocytosis, etc. Then there's all the definitions. Notice she put them all in color coordinated them. She's in Cornell notes. It's well organized, main ideas, summaries. Then she has here the work that she did on cells. Again, all this is in Schoology. If those of you that were absent the last couple weeks, before break because you were sick or you went on vacation or someone in your house was sick, whatever, all this was in Schoology. So it was all in Schoology, is still in Schoology. So you're more than welcome to get it done. You'll see here, these are practice questions, all posted for you to get it done. The difference between, so all these practice osmosis diffusion, what is it? All there. The answers to this particular section were there for you as well as a glossary. And here's transcription translation. She put it together. She actually wrote it electronically, but whatever, and we'll go over those in a minute. And again, I doubled up her work, so I apologize for that, but I think you can see she put a lot of effort into this. We're gonna use our basis for review, and we're gonna answer as many questions as we can this week. Unfortunately, we only have 5 days at half an hour apiece. So, and your final again is next week. So we're going to do our best, but if you're not doing your end, if you're not doing your work, the preliminary work to get it, to practice and go over this material, it's going to be a hard test for you. If you do, I don't think it's going to be that hard. So it all depends on how much effort you put into this. Any missing work that you have, you need to post, you need to get it done. All the lessons are posted on Schoology. All the stuff is either on Schoology or it's on your YouTube channel, or it's on both. So there really isn't a lot of reason that I can find for anybody to have any missing work at all. You've had a month and a half to get that stuff turned in. I have not given you much work on it. I'm recording this, you know, and I'll make it available to your parents and to you as soon as I can. So let's move on.
Mr. Mendoza.
Yeah.
What day is the final?
It's in Schoology, so look on your calendar. I don't remember. It depends on your who you have, which cohort you are. I got to let you know. I suspect we'll be in class this week, but as we discussed before we went on break, COVID was becoming a bigger problem, wasn't it? Yes.
Yes.
Right? And we
Yes.
talked about all the issues that were coming up. I think it was predictable that this was going to happen, isn't it? Wasn't it? We predicted it. I can't believe you're in bed. How are you in bed, Trinity? How do you have your head laid down? Could you sit up, take notes? You have to take notes. It's not, this is a class. It's not something you could just lay your head down, relax. All right, so this is a final, Serenity, a final. You have, this is it. It's not even the first class of the day. It's already 9:16. The sun's up, sun's shining through my basement window. It has to be shining through your window.
Oh my goodness.
All right, so in any case, what we have is, what were we talking about before someone was laying down? Which is, again, one of the reasons you have to have your cell phone, your cameras on, because that's exactly what people do. All right, so you have your review. Your final is next week. It depends on which cohort you are. It's all posted in Schoology when it is. You have a 2-day final multiple choice FRQ. It's all explained in Schoology. Please use that resource. This quarantine, this virtual session was predictable. We predicted it. We said it was probably going to happen with Omicron. We knew it was going to be a problem with the holidays, and it is exactly what we thought it was going to be. We'll see what happens, but we'll probably be back in class on Monday. Then we'll have a final in class. If not, it'll be online and we'll do it here. Either way, you're getting a final, and grades are going to be due either way. So it's going to be important that we get ready for that test, no matter what we do, no matter what you do. All right. So let's take this seriously. So let's go ahead and start with organic molecules, as you see here in the first sheet of the biomolecule. And so biomolecules are organic molecules, which means what? They're made, the molecules that make up living things, but also the key is that organic molecules are molecules that have carbon in them. All biomolecules have carbon. What are the 4 categories of molecules that we've looked at? Everybody should know that already. You should be able to answer it in your sleep at this point. That's carbohydrates, proteins, lipids, and nucleic acids. Right? Those are the 4 big groups. If you don't know that, if there's anything in here that you're like, man, I don't know what he's talking about, then that's something you really need to write down, because we spent a lot of time going over all this. You have to review this material. You have to get in your head. You're not going to be able to use your notes on the test. So what's a carbohydrate? What's a carbohydrate? Can anybody tell me? I'll wait. If we want to spend half an hour waiting, that's fine by me. Less work for me. What's a carbohydrate? Somebody in this room knows. I don't know why there's so much movement in it.
Can you repeat the question? I couldn't hear you.
What is a carbohydrate?
A sugar molecule?
But yeah, it's like molecules that contain sugar.
That's sugar molecules, correct. It is a sugar. Sugars are carbohydrates, not all. Oh yeah, I guess they're all sugars. Carbohydrate is made of carbon, hydrogen, and oxygen only in a 1 to 2 to 1 ratio. Carbohydrates, carbs. Seeing as most of you can't recognize that answer, are not able to answer quickly, assuming you're not incredibly shy, and I know most of you are not that shy because I hear you talk and talk back all day long, so it's not an issue, right, Trenton? Because you talk back a lot. So I'm assuming that you don't know what a carbohydrate is. In that case, you are in big trouble. You have to know what it is. I don't know what else to say to you. At this point, if you don't know what a carbohydrate is, you are in big trouble. You have a final next week, and you're expected to know that a carbohydrate is anything that's made of carbon, hydrogen, and oxygen only, and in a 1:2:1 ratio. So let's take a look at some examples of carbohydrates, the monomers of carbohydrates. Again, this is all review, so I'm going to go quickly. Let's look at a simple one, glucose. Let's compare it to another one called fructose. Two common monomers. And then let's take a look at something called ribose. Glucose has 6 carbons. How many hydrogens does it have? If glucose has 6 carbons, how many hydrogens does it have?
Three.
Huh. I tell you that carbohydrates are 1:2:1 ratio. What is
Oh,
it?
12.
Well,
My fault, my fault.
12. And how many oxygens?
6.
6.
Fructose has 6 carbons. How many hydrogens?
12.
12.
And how many oxygens?
6.
And ribose has 5. How many hydrogens?
10.
10.
How many oxygens?
5.
All right, so that's all there is to it. That's the monomers of carbohydrates. You need to know that they're 1:2:1 ratio. If you see anything that is just carbon, hydrogen, and oxygen, especially in this ratio, you're talking about a carbohydrate. Are we clear? Waiting.
Yeah.
Yeah.
So those are the monomers. Now, interestingly, glucose is a 6-sided sugar. So we draw it like this. Has 6 sides with an oxygen here, and it sticks up. So this is the first 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. And there's actually, that's not right. Sorry about that. I always do that. Mess that up. It's the one that sticks down. So 2, 3, 4, 5, and 6. So here's your 6 carbon sugar. Fructose also has 6 carbons, but fructose forms a pentagon, has one that sticks down and the other one that sticks up. So 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, and 6 prime. So there's, again, 6 carbons. I'm not drawing the oxygens and hydrogens. The odd thing is that ribose has 5 carbons and forms a 5-sided ring. That's 1 prime, 2 prime, 3 prime, 4 prime, and the 5th prime sticks up. This is very important. Ribose, which prime, which number, which carbon is which is very important on ribose. Why is it so important on ribose? What's so important about that molecule, this molecule ribose? Why do I even bother talking about primes? Why is that important? Anyone? I'm waiting.
Because without knowing those, they'll all appear the same. And
Well,
this is just a similar.
that's a good reason. Yes, it's not a bad reason. But the reason that ribose is so important is that there's another biomolecule category that we call nucleic acids. What are nucleic acids? What are the 2 kinds of nucleic acids we've talked about in class forever?
DNA and RNA.
Excellent. Yes, that's right. DNA and RNA. Who said that?
Christian with a K.
Christian with a K. Very good.
Because I'm smart.
Everyone's smart. It's not about smart, but it's about how much work you put in. Everyone in this class is smart, and I agree, you are smart. I never doubt that. I don't doubt any of you are smart. That is not the issue. It is the work you're putting in. That's the only difference between the successful students and the not successful students. So we all clear on that? Everyone in here can do this work. You just either choose to do it or you choose not to do it, guys. That's all there is to it. So DNA. All of these, DNA and RNA, they both, what do I draw this? Let me redraw this. And let me just say, they have a lot of stuff. RNA and DNA have a lot of things in common, and they have some things that are different. So let's try to look at what's in common first. Both DNA and RNA have a 5-carbon carbohydrate at its center, 1', 2', 3', 4', and 5'. All of them have this structure. Both DNA and RNA have that. What is the difference in the sugar between DNA and RNA? Between deoxyribose and ribonucleic acid. What is the difference between the two?
One doesn't have ribose and
No, they both have ribose. Listen to the words. Deoxyribose.
one doesn't have oxygen in it.
One doesn't have oxygen is correct. And so in deoxyribose, so this is where they have in common, right? In deoxyribose, what you have is you don't have, well, that looks anemic. At this position here, you have a hydrogen. At the second carbon, you have a hydrogen. So 1', 2', 3', 4', and 5'. In the second carbon, you have a hydrogen. In ribonucleic acid, you have unbelievable. Sorry, guys, give me a second. In ribonucleic acid, you have at the second carbon, you have an OH group sticking off this side. So there is, oh, is it time? Is it 9:30 already? 9:29? Are you kidding me? Wow.
Time to go.
No. Oh, nice. Who's that? Maya. Oh, you're so in such a hurry to go, Maya. So sorry to hear that. No, we still have 10 minutes left. So when we're talking about the difference between RNA and DNA, RNA has an OH group sitting here. DNA doesn't. So this is deoxyribose. This is ribonucleic acid. So you have to know the difference between the two. That's important. I will be testing that. Then when you look at 5', they both have sticking off the side this phosphate group. They both have this phosphate group. Why is someone joining now with 10 minutes left in class? So there's a phosphate group here. Jesus. So the phosphate sticking off to the side there at the 5' end on both of them. And then they also both have a nitrogenous base sitting off to the side as well. Either a double ring or single ring nitrogenous base. So these are what we call nucleotides. These are monomers of nucleic acids. So we can take these and we can repeat them over and over again into a chain. So we can take this nucleotide we take a bunch of these and put them in a chain from 5' to 3' over and over again. And this would be a chain of ribonucleic acid. How do I know it's ribonucleic acid?
Because it has OH.
Because it has an OH here. I know, so if I give you something like this and I ask you is it DNA or RNA, you should be able to tell me it's RNA, right? Because it has an OH here. We call this a phosphate sugar backbone. Both DNA and RNA have a phosphate sugar backbone. Why do we call this a phosphate sugar backbone? Who can answer that? So uncomfortable when people this is always so uncomfortable when people don't prepare. What is a phosphate sugar backbone? Why do we call it phosphate sugar backbone? Look at the picture in front of you. Phosphate sugar backbone. Why do we call it phosphate sugar backbone? Serenity, Heaven, Bailey, Precious.
Because they're all lined up. I'm just looking at the obvious.
Well, it is obvious. The answer is incredibly obvious. Phosphate sugar backbone. Why do we call it phosphate sugar backbone? What is the deal?
Is it because like the phosphate is sticking out the back of the thing?
Yeah, there's phosphate. That's correct. And what's this thing here? This is a phosphate. That's correct. What is this thing? Nobody knows. I just said it. Nobody's listening to me. I'm wasting my time.
Sugar.
Sugar is correct. This phosphate sugar, and it repeats phosphate sugar all the way down. We call it a backbone because it looks like a backbone, looks like a vertebrate, a spine. So it's a backbone, and we have phosphate sugar all the way down. And then you have your nitrogenous bases here on the other side. What are the nitrogenous bases? What are the nitrogenous bases in RNA, for instance? Do you hear this silence? Do you hear this? Oh,
Is it ATGC?
very good, very good. It's AUGC in RNA. But correct, absolutely. Who said that?
Liberace.
Yeah, so that's in RNA. In DNA, the nitrogenous bases are A, T, G, and C. So the only difference between DNA and RNA is structurally is that the OH is not there in DNA, and that in DNA you have a T. These nitrogenous bases could be a T, where in RNA there is a high OH group here, and that there can only be a U, AUGC. There can be no T in RNA. So everybody should know that much so far. Another difference is that RNA is a single strand, single-stranded. Where DNA is double-stranded. That's another big difference. Now, RNA can form different shapes, though, even though they're single-stranded. They can form these shapes that we call rRNA can form ribosomes. That's what the R stands for, is ribosomes. They are the enzyme that put them together amino acids. You should know that. That's going to be on the test. You should review that. You should watch it. You should have your notes. I don't know what else to say. Then you have another possibility, which is another tRNA, which stands for transfer RNA. In my opinion, looks like a T, which the ribosomes are the enzyme that puts together amino acids into proteins. everybody should be writing that down since you obviously don't know the answers to these questions. I suspect some of you are not even writing. I think some of you are in bed, but that's fine. I hope your parents look at this video so that they can see that the level of preparation that is or is not happening. tRNA carries the amino acid, all right, to the ribosome. And then the last type of RNA that we've talked about is mRNA, which is single-stranded and it has a cap, a 3' cap, and it has a poly A tail, and that carries the message. Carries the message from the nucleus to the ribosome. So those three RNAs together, they help in the process of translation, right? Of making taking the message in RNA and making a polymer of amino acids. The double-stranded DNA, it's double-stranded. It forms a double helix, an alpha helix. So it forms something like this. It's a double strand. So there's two strands. There's a 5' to 3', and here's a 3' to 5'. They're anti-parallel. They're double alpha helix. Alpha just means the shape of it, and together they form the nitrogenous bases, form hydrogen bonds between the two strands. And these hydrogen bonds help keep the two strands together. They're weak bonds, and wherever you see an A on one side, you're going to have a T on the other. You have a G on one side, you're going to have a C on the other, because that's how they match. The A matches with the T, the G with C, we call that base pairing. And that's really the big difference between DNA and RNA. DNA is found only in the nucleus. RNAs are found everywhere in the cell nucleus where they're made. They're made in the nucleus, and they're found mostly in the cytoplasm. And endoplasmic reticulum. All right. So that's where you're going to find RNA in the cell. So that's pretty much nucleic acids, everything you need to know for the test. Of course, there's more stuff there in the review packet. We're not going to be able to cover it all if you don't do that work. You're not going to be able to be ready for the test. The final's coming. This is just one of the biomolecules. Then we still have osmosis, diffusion, transcription, translation, gene expression, organelles, cell structure, membrane transport. All that stuff still is coming as well. We only have 4 days to do all that, and we only have half an hour a day. So if you're not doing your own preparation, you're not going to be able to do as well as you could have on the final next week. All right, that's it for class. Have a great day. I hope you participate more tomorrow. Hicks, are you still there? In school, is that something that the community is going to start arguing now, or do we all agree on that? Yeah, so
I agree.
yeah, so
But school home is kind of better.
what'd you say? School at what?
Being at home is better.
That while you're in school, or being at home without school?
being at home in school.
Oh, you think that's better? Well,
Honestly, for me, I feel like certain classes I feel like I need to be in school, but like some classes like algebra and like biomedical, so like I can be at home for that type of stuff.
well, I think that I guess you're right. It depends on the class and whatever. I guess the part that I miss is I miss us all together. I miss watching you guys interact with each other, watching, Marshall come in and plays games, watching Frank annoy everybody on purpose. Frank, it's on purpose, Frank. It's on purpose. Yeah, just quietly sitting there trying to be like, just be, but nobody lets him be, especially me. I miss the evil looks that certain students give me, even though they don't turn their cameras on now. That's the kind of thing that you kind of miss when you have that interaction, that social interaction where you have it doesn't feel supportive necessarily on a day-to-day basis. But having that routine, having those people in your room with you, knowing who they are, even if they annoy you, is a lot better sometimes than that uncertainty that we get when we're at home. Like I'm at home, I'm in a comfortable place. I set up my little cave down here very comfortably. I have my video game playing in the background, waiting for you all.
Did you get some yet? Oh, my bad.
What happened?
My bad, Mendoza. I was talking to my sister. I didn't know I was off mute.
All right, no problem. So I just think that I miss that social interaction. And on top of that, even if the class is something that's a little lighter on the and thinking or what have you, I think it's better to be in school because you can stay focused. You don't have your sister talking to you. You're not distracted by other things. I had a kid today that was watching the news. His parents or grandparents were watching the news while he was trying to do class. And even now, look at Tyler. I think she's having a conversation with somebody. It's not me. So is she trying to get her French fries or something? Is that what's going on? Is it lunchtime for you, Tyler? Is that what's going on? Can anybody tell what she's doing? Is she talking to us?
I think she listening. She's talking to somebody else. I don't know.
Anyways, the point is that having an interaction in class and being focused on material in class is a lot better than what we're doing here. But it's what we have. And next week, hopefully we'll be in class. Now I got to say, I just read an article this morning on how much more severe Omicron is for students, for young people. In fact, we're putting about 633 young people in the hospital every day in this country. So it is a concern that opening schools. I know New York's decided to open schools, and they decided that because of how bad this virtual stuff is for us personally, for our emotional stability, for our learning. We know that this type of learning is not the best. We're separated from each other and we're not focused. So we know that this is not great for us as human beings, but there's a concern about health and safety. And so with 633 kids, and I say kids, I mean young people from 0 to 8 to 22, I think it is the age range, are going into the hospital every day because of Omicron, or Delta didn't impact young people as much, but Omicron is impacting young people. So it is a concern. And so I'm not sure where we're going to be next week. My guess is we'll be in school. However, no matter what, guys, your final is next week, whether it's virtual through Schoology, and I will verify that it works before you even log on. And if it takes us our planning time to get it done because it doesn't work during class time, whatever it takes, if we're still at a half an hour break, half an hour per class, then we might be testing all week, because instead of two 1 hour exams, you might have 4 half-hour exams. But we're going to get this exam done. It's your last major grade. It's a giant grade. It will impact your grade, good or bad. For some of you, that's a good thing. For some of you, it's going to be a bad thing. It all depends on your individual breakdown in your categories. Monday's the last day I'm grading any late work. That's it. If it's not turned in by Monday, you can turn in, but I'm not grading it. And also Monday, the test review is due. The final review is due on Monday. It says, it's labeled test review. In Schoology, all the assignments are there. They need to be done. Now, those of you that did it over the break and turned it in, you got your 1,000 points, so you'll get them in a minute now. And think in your class, let me see. I don't know how many people turn it in your class. But if 1,000 points go missing, that's going to be harder for you to get a decent grade, guys. But it's going to be what it is. I got 1, 2, 3, 4, 5 people turned it in this class. 6 people turned it in this class. And I graded one of them already, and they got their full 1,000 points. Everyone else, that's due on Monday. So you need to get it turned in. In fact, let me go ahead and change the due date on that assignment right now, so I don't forget. But this is due on Monday now for everybody. You had the option to do it over break. Yeah, that would have gave you a month to do it. I think it would have made your life a lot easier, as I told you last week. But now it's due on Monday for everybody. So it was excused, but now this is due on Monday. Is that clear to everybody? Nobody's confused about that? I can't hear you.
Yeah.
All right.
Yeah.
It's due on Monday. That gives you a week to get it done. We're going to go over some of the material here in class. All the class lessons have been posted on YouTube. You have all your virtual labs are still available. Anything that you've missed you can still turn in. I changed it, so now it says due on Monday, and that's it. There's no everything will be excused, or if it's not done, because this is now mandatory. So I understand some of you, I'm just reading some messages and some of you sent recently. Some of you don't understand, is that right? These concepts we're talking over? Hello? I'm talking to all of you.
Yeah.
So you say that it's difficult. There's a couple issues I want to talk to you about, and there's something really where you have to be reflective on. Okay, and everybody in this class is different. Some people obviously understand it and they're fine. Others don't get it and they're lost. I got to tell you, I can pretty much guarantee that most people that don't understand anything or don't understand what's going on are people that are not doing the work on a day-to-day basis. And they're not focused, they're distracted, they're absent. I don't want to name any names, but they are who they are. I don't think this stuff, and by the way, could everybody go into the chat and put your first and last name so I make sure I have attendance taken care of? I don't have to worry about attendance, please. Hold on, there's something going on. I'm just trying to sign in with my iPad, and for some reason, it's giving me a... There I am. All right. So when I nag you on a daily basis to please pay attention, to take notes, to take Cornell notes. When I tell you to do that, it's not for me. It's not because it's going to help me. You're not doing it for me. So get that out of your head. Those activities are so that you understand it on a day like next week when you have to do a final. You got to be honest with yourselves. You have to get in these habits of doing the right thing on a daily basis so that you can be successful in high school and then in college. You can't be successful if you're not doing it on a daily basis. So I got to say, you know, this stuff of not understanding, saying at the end, I don't understand stuff. I respect that. That's acceptable because that's going to happen, especially when you're trying to remember things that happened in August. But that's why you have your notes, ladies and gentlemen. You have to get into the concept that when I tell you something, I'm telling you from 20 years of experience. I'm telling you from as a teacher, as someone who's educated in this. I'm not telling you because I'm nagging you and I want to bother your life. So, all right. So if you need help, let me be clear. I'm gonna explain some things here. I'm gonna tell you these are things that are gonna be on the test, on the FRQ or on the multiple choice. If there's something you don't understand, you need to make a note. I don't understand this. And then you come and see me at 1 o'clock, because at 1 o'clock, I'm going to be online like I was yesterday. And no one showed up, by the way. So people that don't understand, that say they don't understand, I respect that. What I don't respect is you don't understand, but you don't show up to open office hours. You don't go to tutoring, right? So if you want to get better, you want to understand, put the effort in, right? I respect not understanding. That's normal. It's not easy stuff. But if you don't understand it, you got to get help. You have to figure it out. Don't sit back quietly. Ivy, are you there? I can't see you. You're the only person that I know that doesn't. Wait, Tyler, your thing's off too.
Yes, I'm here.
Okay. So don't sit there quietly and, not pay attention, play games and jokes or roll your eyes or go to the bathroom 3 times a period.
Who playing games?
Not you, Ivy. I'm talking about the guys. So when you're doing that on a day, every other day, or you're not focused, and then you're at the end of when it's time for the final, you're like, I don't understand. Well, no duh. That should be expected. It's a predictable outcome. Just like us being virtual was predictable, and we predicted it, didn't we? Didn't we predict that this could happen?
Yeah.
Then it should be predictable that you're not going to have understanding if you're not putting the effort in on a day-to-day basis. There's no doubt this stuff that you're learning in high school and then again in college or wherever you end up after high school, it's not going to come to you naturally. There's not some magic that happens. There's no spell that you can cast to get you where you need to be. You have to put the effort in, consistent effort. And the less you've done in the past, the more you have to do in the future. That's all there is to it. All right. Let's go ahead and do this. We covered yesterday. Are there any, here's my asking right now, I'm asking, were there any confused points over yesterday where we went over carbohydrates, we went over nucleotides, we talked about it yesterday. Are there any confused points?
I had to step out yesterday and I missed the part where we talked about nucleotides, but I heard the carbohydrate part.
Wow, you missed the whole nucleotide conversation. Okay. What about anyone else? Are there anything that you were confused about yesterday?
Yeah, I missed the nucleotide part too.
So ribose and deoxyribose, are those sugars?
That's correct. This structure right here is a sugar. It's a 5-carbon sugar, so it's called a pentose. There's sugar, pentose. Notice I'm writing while I'm talking, not reviewing a pre-recorded video, correct?
Yeah.
All right. So it's a pentose. This pentose, this thing called specifically called ribose, is the part of a nucleotide. That is not a nucleotide. It is a sugar, but it's part of a nucleotide. The nucleotide is made up of 3 parts. It has a phosphate group attached to the 5' carbon. 5' carbon. This is attached. This is 1', 2', 3', 4'. They're just numbers. The little hash mark is a prime. This part is a carbohydrate, a sugar. This is a phosphate. What's a phosphate? It's not a sugar. It's a phosphorus with 4 oxygens. That's what a phosphate equals, a phosphate. So when you look at a phosphate, it's 4 oxygens attached to the center phosphorus, and that together acts as part of the backbone of DNA and RNA. Off to the side, off this 1', you have a nitrogenous base. The nitrogenous base can be a single ring or double ring. Single or double ring. A single or double ring that can be one in DNA, one of 4 different nucleotides, nitrogenous bases, and in RNA, it can be 4 others. What are the 4 that we're talking about?
A, T, G, and C.
A, T, G, and C on DNA, or what about RNA?
A, U, G, C.
That's right. So those are the 4 that could be in RNA. So that's pretty much all there is to a nucleotide. But then nucleotide can form in, I'm not going to review the whole thing that I went over yesterday. I'll post that as soon as I'm done editing it, and you guys can look at it at your leisure. But I am going to talk a little bit about DNA, since that's what we're going to go on today, is the gene expression. So if you take this nucleotide, again, a ribose sugar, and DNA means that you're not going to have an oxygen there, it's a hydrogen. And you're going to have your nitrogenous base. It could be any one of those 4, A, T, G, or C. It could be double ringed, or it could be single ringed, whatever. And you're going to have a phosphate. So that is your nucleotide. You're going to have a bunch of these, and they're going to be connected. They're going to be connected. 5' to 3', 5' to 3'. They're going to continuously be connected in that way. Just like we did in class, just like when we made the paper mache, the cardboard model, just like when we work with the precasted plastic models. We went over it in class. We talked about it. Everybody said, yeah, we understand. So there we have it. That would be a DNA, half a DNA molecule, right? Because it's only, because a molecule is what? What is DNA? Single or double stranded? Double stranded. So we need a second strand in order to have DNA. Now these 2 are going to be, obviously I hope you understand they're much longer than this. I'm just drawing this because it's a billion nucleotides would be too much to handle at this point. So I have these 2 nucleotides. How are they going to hydrogen bond? Where do the hydrogen bonds occur?
At the nitrogenous bases.
That's right, at the 2 nitrogenous bases. How can we get hydrogen bonds to form here? How can we get them to form? Can they form this way?
Turn it.
That's right, we have to turn it. We have to turn it upside down. Now they can hydrogen bond, can't they? Now you can form a bond, hydrogen bonds, which means they just attract each other like magnets. in DNA. And in DNA, A and T have 2 hydrogen bonds, and G and C have 3 hydrogen bonds. They just attract each other like magnets between the two, holding the 2 strands together. But please notice that on this end, you have, let me do a different color. On this end, whoever's co-host, let them in, please. On this end, you have a 5 prime, and on this end, please notice, you have sticking up, you have a 3 prime. So where this end has a 5 prime sticking up, this end has a 3 prime. Also notice at the other end, this side is your 3 prime, and this side is your 5 prime. So do you agree that they're opposite? When you guys have TVs on in the background, I understand that you can't control that often when TV, because you can't control what happens in your house. But when you have all that background noise happening, television, music, whatever it is, you're distracting yourselves. I'm telling you, you cannot do 2 things at once. You cannot listen to 2 things at the same time. So don't be surprised if you leave this session with less understanding than someone who's not eating ramen noodles, someone who's not listening to the news or commercial, someone who's not listening to music or whatever else is happening. Just don't be surprised at that. So these 2 are what we call anti-parallel. They're anti-parallel. And what that means is that even though they're parallel, they're one next to the other, right? They're opposite. This is 5 prime to 3 prime. This is 3 prime to 5 prime. It's the only way they can be hydrogen bound to each other. If you remember making the cardboard models, you remembered you had to give it all kinds of twists and turns to make it work. Well, that's why you did that activity. So you got that in your head that you have to twist and turn it in order to get these 2 to hydrogen bond. So once you get that, then you end up with a cell, right? Let me make it even bigger. End up with a cell. How
The
much?
genes are in that spiral, right? They're in that spiral. That's the 3, 4 bases connected.
That's correct. That's correct. That you got it. Someone's gonna have to keep track of time, please, so we don't go over. I don't want to hold you up. So you know that the nucleus, in the nucleus, you know you have DNA, and DNA has 2 strands, right? Just like just exactly what we just looked at. So again, 5 prime to 3 prime, 3 prime to 5 prime. So there's 23 copies of these things. We call them chromosomes.
Time to
23
go.
pairs. Say again.
It's time to go.
Oh, bummer. Well, we'll have to finish this tomorrow. So if you're confused about any of this or any of the stuff from yesterday, see me at from 1 to 2. I'm gonna be online. You guys need to get ready. Do that review for the packet for the test. Gabby, let me talk to you before you leave.
Oh, I was gonna ask if I could talk to you real quick.
All right, let me stop recording. Imad, I need to see your face. That piggy bank is not good enough. Taylor, again, camera on, please. I need to know you're in the room. I have people in bed, laying down. Putting the camera up so you can lay down in bed is not sufficient. You should not be sitting in bed. I will be having conversations with parents if I can't get you guys to focus and take notes. Marshall, what's wrong with you? I don't know. It's such a confusing life. But you should be taking notes. Do you have something to take notes with, Chloe? Chloe Hart. All right. All right, is this sharing yet? I don't know. I need you to participate verbally as well. Everybody should see the screen now. I'm still waiting. Imad Sumrain.
Yeah.
Turn your camera on or I'm calling your mother.
I will, I will, Mr. Mendoza. Give me a bit.
All right, so let's go ahead and start to go over what's going to be on this test. This is what one of your fellow ninth graders completed. She completed this over break. She said it was not really that hard and that it took her just a few pages a day and she finished over break. You've had this for about 3 weeks. You'll have it by the time you take the test, you'll would have this for a month. And we're going to go over this content, but this are her answers. This is what this is wrong here. There were several pages of this. Unfortunately, I inserted the AP review in here as well. So I'll have to post the other review in later. But in any case, the bottom line is there are several pages. Several people have done this, have completed it. So they're ready to go. Imad, if I put you out, you will not be able to get in. I'm just giving you that warning. All right. So when we talk about organic molecules or biomolecules, they're organic molecules.
I'm trying. I'm sorry.
It's fine. Organic molecules are made of carbon. You should know that. The organic molecules are made of carbon. There's 4 major biomolecules: carbohydrates, proteins, lipids, and nucleic acids. And so let's talk about these first. Organic molecules, as I said.
Got it.
As I said, organic molecules are made of carbon. Those 4 biomolecules are the big categories of organic molecules. You expect that if you're looking at a biomolecule, there's going to be carbon involved. I hope that's an expectation everybody has. From organic molecules, you go to organelles and then the cells. From cells, you go to tissues. From tissues, you form organs, and organs form organ systems. Let's take a look at carbohydrates.
Wait a second. What's after organic molecules?
Organic molecules can form organelles. When they come together, they form organelles. Then organelles come together to form cells. Right?
Thank you.
All your this review for this test is posted in Schoology if you did not pick up a hard copy of it. So you should make sure you're doing the review. Remember, this final is worth a lot. Some of you have decent grades. You won't have decent grades if you don't do well on the final. Maybe it'll be better, maybe it'll be worse, but well, it all depends on what you do on that final. It's a big part of your grade. This is the final for the semester. The semester grade is going in after next week. If we're still virtual, I don't think we'll be virtual. I think honestly, the odds are we're going to be in class based on what I've been reading, but we could still be virtual next week. If we're virtual next week, I don't think we're going to be half-an-hour classes. If we're virtual, you're still having your final. If I have to break the final up into 4 parts, you're still having your final. No matter what happens, you're having your final next week, one way or the other. So make sure you're ready. All right. Carbohydrates. They're made of, oh, let's just start it. Oh, I can't. Carbohydrates are made of what? Let me just start writing again. What are carbohydrates made of?
Sugars.
Sugars are carbohydrates. That's correct. But carbohydrates can be monomers, they can be polymers, they can be disaccharides, they can be polysaccharides. So what are they? Well, the base unit is CHO. They're made of carbon, hydrogen, and oxygen. They have a specific ratio. Does anybody remember what the ratio is?
1 to 1.
That's right. 1 to 1, 1 to 2 to 1. So if I have a molecule like, let's say, glucose, and I have a molecule like, let's say, fructose, both of these have CHO, both have CHO. They both have 6 carbons. How many hydrogens do they have?
12,
12. And how many oxygens do they have?
6.
Correct. So they have the same basic formula, but they're different. And how are they different? When they form a ring, one's a ketose and the other's an aldose, by the way. That's the big difference. But when they form rings, they form structures like this. Essentially, one forms a pentagon, the other a hexagon. So this has the 1' here, 2', 3', 4', 5', and there's a 6. There's a 6th one sticking up. 6'. Here you have a 1', 2', 3', 4', 5', 6'. So you still have the same number of carbons, they just have different shapes. One's a pentose, the other's a hexose. Now I say this because there's another one we talked about called the ribose. Notice they all end with what?
Ose.
Ose. Ose means sugar. Ose means sugar, carbohydrate. I should see people writing things down, including you, Miss Mutated SpongeBob Taylor, if you're still there. But I can't tell because I don't see you.
I disappeared.
I need to see, you need to reappear. Otherwise, there'll be a I will be appearing at your house to have a conference with your parents. I can also disappear and reappear.
Good on that.
I have
I'll just
this
stay
thing
here
called
then.
a car. I can drive anywhere in the city. All right, so ribose. Let's take a look at ribose. It's one of those important molecules. One of those things that sticks out. We know there's a 1', 2', 3', 4', and 5'. What do we know about ribose versus another molecule that's similar, that's deoxyribose?
So these are all sugars, which are carbohydrates.
These are all carbohydrates. I see the OSE. OSE. What's the difference between deoxyribose and ribose? Deoxyribose.
The amount of oxygen?
Correct. The amount of oxygen is involved. So does deoxy have extra oxygen or less oxygen?
Less.
Less, that's right. And where is deoxy missing an oxygen? Does anybody remember?
2.
That's right. It's on the second carbon. It's missing an oxygen. It's just a hydrogen. All the rest have the ribose has all its oxygens, and in other words, this one here, this deoxyribose, I don't know why it doesn't. Something's going on with this. It's at 81%. So this deoxyribose is not C6H12O6, it's C6H12O5, where ribose is C6H12, I'm sorry, not C6, I'm sorry. It's C5, remember this is going to be what? If it's C5, it's C5H10O5, where this would be C5H10O4, right? Because it's missing an oxygen. So in any case, that's not really what's important here. What's important is that the difference between ribose and deoxyribose is this oxygen. Here's an H, where here would be an OH. Right, so that's a big difference. Now I'm going to switch gears and look at something else called the nucleotide or nucleic acids. It's another one of those groups of carbon compounds, organic molecules that help form life. We call it a biomolecule. There's 2 kinds of nucleic acids. What are they?
DNA and
DNA
RNA.
and RNA.
That's correct. DNA and RNA. DNA has at its base which one of these two, the deoxyribose or the ribose?
Deoxyribose.
Deoxyribose, that's correct. So this does not have the hydrogen on it. Still has, it's ribose, so it only has 5 carbons, where this one has, the RNA has that hydroxide group on it. Again, I don't want to keep labeling them, but there's a carbon 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. And the same here, 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. Now, both of these on the 5 prime, they have a phosphate. They have this group of atoms called a phosphate. I want to remind you that phosphate is actually phosphorus. Oops. It's actually phosphorus with 4 oxygens on it. Is that clear? Yes or no?
Yes.
Yes.
Yeah.
Excellent. And then here off to the side on both of these, they have these things called nitrogenous bases. It's either a single ring or a double ring. It's coming off the first carbon. Single or double. What are those nitrogenous bases? What are the possible nitrogenous base side chains for DNA?
A,
A,
T, C, and G.
G, T, C.
Correct. Excellent. A, T, G, or C is correct. What about RNA?
A, U, G, C.
Perfect. That's correct. So that's pretty much it, except how they're put together. When we look at this guy here, this DNA molecule versus this structure here, when we look at this guy, we can put these guys together like this, like that, like that, like this. And we can put these guys together here. We could take these monomers and we can form a what?
Polymer.
Polymer.
Polymer, that's correct. We can form a polymer. We can do that. And when we make this polymer, that's what we think of when we think of DNA and RNA. Now, when you're talking about RNA, you're talking about it's single-stranded. But it still can do hydrogen bonds. It can still form all kinds of shapes. So it can form this little shape that looks like this in the form of rRNA. And we call this a ribosome. It acts as an enzyme that connects amino acids together, makes connections between amino acids. All right, that's what ribosomes do. There's 2 subunits, a large and a small, and rRNA is the main component. Even though they're single-stranded, they can come together and form these structures. Another single-stranded object that's formed when it bends and it attracts itself is this other thing called what? What is this called?
tRNA.
tRNA, excellent. tRNA, and tRNA transfers the RNA, carries the amino acids to the ribosome. All right. Another form of single-stranded RNA is this mRNA that we know has this poly A tail on it, that has a 3' cap on it, that has the introns taken out and the exons are spliced together, but you make this RNA that carries the message to the ribosome. From DNA, right? So when we look at these different kinds of RNAs, they all have these phosphate sugar backbones. They all have these nitrogenous bases.
Can you zoom in a little bit on mRNA?
Sure. That carries a message to the ribosome. It's a little messy. I'm sorry. I'm trying to write while I'm holding it. No. All right, is that readable otherwise? It's not neat, but is it readable? Good. All right, somebody want to let her, Sophia in? I don't know why Sophia left.
Her computer died.
Her computer died.
I hate that. All right, so
Mine
that's,
might as well. Sorry about that.
we should have it plugged in.
Yeah, I'll
Ugh.
try.
Wait, so the mRNA carries like the codons
That-
to the, and then after you translate those to like a protein, the tRNA carries it to the ribosome.
Let's go over that tomorrow, okay? But you're close. You're very close. You're just clarifying some misunderstandings there. So you're very close. So the ribosome is the enzyme. It needs a message to make the amino acids, and that's what the mRNA does, is the message. The tRNA carries the amino acids. We'll go over that again tomorrow. But this over here, this DNA is double-stranded. And it forms a double helix. And this is, of course, in double alpha helix. It looks something like this. In prokaryotes, it's circular, but in eukaryotes, it's linear, where there's 5' on one end and 3' on the other, and there's 3' on this end. So they're anti-parallel. I don't want, let me make sure I get this straight. So on one side, it's 3' and the other side is 5'. So they're anti-parallel. They match up and they're held together by hydrogen bonds between the nitrogenous bases. About 3.5 bases per turn. We built this. Didn't we build this in class, right? We cut it out, built it in paper, we looked at it with a prefabricated 3-dimensional model. So we built it many different ways, but the double helix is the nitrogenous bases attached to one another, attract each other, and the way they attract is A is attracted to T and G is to C. They have G and C have 3 hydrogen bonds, A and T have 2. They cannot fit any other way, so that's why A and T go together and G and C go together. In RNA, the same thing happens, except you don't have a double helix. There's no 2 strands, it's only 1 strand, but they still kind of do all kinds of twisting and attracting to one another. And of course, when you're doing base pairing in translation or even in transcription, you're going to do this A goes to U and G goes to C kind of thing. All right. So that's DNA and RNA, all based on the ribose sugar at its center, isn't it? They all have ribose sugar. We call this a phosphate sugar backbone. Why do we call this a phosphate sugar backbone?
Because the phosphates all connect.
That's right. It goes from phosphate to sugar, all the way down. And it looks like a spine, right? It looks like the spine of a book. It looks like the spine of like a vertebrate. These, if you use your imagination, you can see this is a vertebrate, this is a vertebrate, this is a vertebrate, and they all form a spine. So we call it a backbone, phosphate sugar backbone, and the nitrogenous bases stick off the side. That's what this black line is. This black line is phosphate sugar, phosphate sugar, phosphate sugar, and the green bases stick out from each of the backbones, sticking out and hydrogen bonding to one another. Here again is the sugar phosphate backbone, and the hydrogen, the nitrogenous bases all stick out to from the inside towards each other, attracting each other and making a certain shape. In this case, it's a T shape. In this case, they form an enzyme. And in this case, they're waiting to match with the ribosome so that it can be read and turned into an amino acid chain. So that's what we're dealing with here. Are we good with that so far?
Yeah.
Is that a decent review? Nobody has any questions or concerns?
Oh yeah, it was pretty decent. Thank you, Mr. Mendoza.
Well, we still got 5 minutes. I think we're done at 12. Aren't we done at 12? Somebody have to help me out. I don't know.
Yeah,
We're done at 12.
Yeah, it's 12.
I do have a question about the packet that you gave for before over break.
Yeah.
Because I wasn't there to get it because my dad didn't want me to come to school because of like the COVID stuff. So
Am I
is it
recording
like
this? I just want to know if I'm recording this because I'm about to.
you are?
I also wasn't there, so. I also
Is
didn't
it?
pass
It's online, right?
I posted it on Schoology, girl.
Okay.
I said that in the beginning of class.
Okay, I was just making sure.
Oh my, okay, but that's why I'm bleeping over here. If you can't find it, talk to me tomorrow, okay? Or during school, today we have, I have office hours right after this, okay? So make sure you check with me if you can't find it. It should be all in Schoology, all the assignments. And this is, what we just talked about was based on this, which as you see, Jasmine did over break. And we went over first period, as you can see. We did pretty much the same thing. And
Do you think you could continue doing it like this? Like instead of like you just playing the video like you do in class, could you like actually write it? I think it's better for me when you like write it with us.
yeah, okay, I'm fine with that.
Yeah, I like that too. It's easier.
I agree.
It's easier for you,
Yeah, it's easier for me.
selfish children.
And I can understand
It's
more.
easier
I
for us to
understand it,
understand.
but, like,
Rightfully
this
so.
is-
Yes, we are selfish.
Exactly,
like I
because you do it at the same time as us so that we all on pace and you're not just skipping through the video.
Ooh, I want to say
Right,
something.
you're trying to take the lazy way out.
Oh, you coming for me? Are
Good
you?
job.
Yeah.
It really be given.
Can you zoom out
Just
more?
watch the video. Don't. I'm not gonna.
I
just
hear
watch the
you.
video.
That's fine. That's fine. I can write it in class. It's not a big deal for me, really. I just like to record it. I'll post whichever one I do best. That's fine. Whatever you want. Are you good?
Can you zoom into the stuff that's like higher up there?
This stuff here.
Yeah, like but like go down. Yeah. Okay.
So it goes like mRNA to tRNA to rRNA.
Yeah, we have a few minutes. I'm going to go ahead and do that then. The way, what you're dealing with here is you have the nucleus. The DNA is in the nucleus, right? Only in the nucleus. And there's genes on the DNA. There are these areas that we're going to express. We're going to express these things. That means they're either going to make some kind of RNA. So when the first thing you're going to do is you're going to these genes, these green things are genes. They're going to be expressed. And how do we express them? We have this enzyme called RNA polymerase. There's three kinds, 1, 2, and 3, but that's fine. RNA polymerase come in and it's going to read It's going to turn the DNA into what?
RNA.
Into RNA. That's right. Where it reads, it's going to read. Let's go ahead and take a look at it. Let's do one of these things. Let's take a close look at it. Where it sees A, T, G, C, T, etc. Right? Continuing. It's going to put, instead, it's going to make RNA. So it's where it sees an A, it's going to put a what?
U.
U, A, C, G, A, right? Etc. So now you have RNA. Now you have RNA. Now RNA.
This is transcription, right?
That's right. This is called transcription. That's correct. Excellent. So this is transcription. And where you see, and still all happening in the nucleus, is all in the nucleus. So now you have RNA. Now there's three ways this can go. Maybe this gene is going to be made into a protein. If this gene is meant to be a protein, it's going to go through something called, it's going to become ultimately an mRNA, which means that there's going to be sections taken out, exons will be spliced together, introns leave, exons get put together, you get a poly A tail, you put a 3' on there, 3' cap, you're done. That becomes an mRNA and it goes into the cytoplasm. That means outside the nucleus, which then could go either into the RER, if it's going to be a protein that has to go into an organelle, into the membrane or outside the cell, or it's going to be made in the cytoplasm into a protein. That's translation. We'll talk about that tomorrow, probably. Then the other thing that can happen in the gene is it can take that RNA, it can become a tRNA. Or that gene could then become an rRNA. Man, sometimes I'm so messy with my drawings. It wouldn't be so bad, except you guys pick on me all the time about it. I have to hear Mod make fun of me all day long. So anyways, you can have that gene can become an RNA, a ribosome. The rRNA leaves and becomes a ribosome out here. So ribosome looks like this, a small subunit, a big subunit. Or it can become a tRNA, and if it's a tRNA, it's going to go and pick up. It's going to go and pick up an amino acid. Maybe the amino acid is methionine, and this that has an anticodon up here. That corresponds to a message on the mRNA. This is the mRNA. And maybe on the and then maybe you have AUG here. Oh. AUG. And here you have UAC, right? So then what's going to happen is the anticodon is going to find the codon. There's going to be the first amino acid. This is the start codon, right? The first amino acid in the sequence. And the mRNA is going to be read. And it's going to be read, and, you know, maybe 3 at a time, because there's a triplet code. G, G, C, A, U, C, U, C, A, etc., right? It continues. Every 3 is going to be an amino acid. And that amino acid is going to form a chain of amino acids that we call a polypeptide. So that's the short of it, right? That's a short summary of what you should have done over practice during break, but what you have to do now if you haven't done that, and all that is posted in Schoology already. Is that clear to everybody? So that
Yes.
when you get that work done, as much of it, start with the stuff that you're most confused about and ask me questions during office hours if you need help or if you're confused about something. Obviously, you want to turn in all your work, get all your work done that you can, because grades will be due next week and class is over, ladies and gentlemen.
Bye.
Bye, children. Bye-bye.
Mr. Mendoza,
Yeah. Yeah.
so I ran downstairs to charge the laptop so that it would last through the extra 10 minutes. And I was ready to I was like to my mom, I was muted. I was like, you got to vouch for me.
All right. First of all, does your mom know you have a pig as your symbol?
Yeah.
I'm going to start we're going to have to go. I'm going to have to go to Saturday prayer for you and pray for your soul, boy. How you not eat? Go ahead.
I think the pig is pretty funny.
It is funny, but if you're not allowed to eat it, why do you got it as your symbol?
Well, you see, it's not just any pig. It's a Ukrainian pig. See?
Wait, well, why is a Ukrainian pig? Wait, what?
See, this one represents the Ukrainian debt crisis.
Oh, I see. That's
Yes, because it's a piggy bank.
You are so smart and just such a smart aleck. It really drives me insane. I think I have to be online now. Let me check. I think I have office hours. Pretty sure that's the case. I should print this schedule. No, we have lunch now from 12 to 1. And then from 1 to 3 is 8. From 1 to 2, I'm online for you guys.
Okay, I'll see you later, Mr. Mendoza.
All right, take it easy. Have a great day, kiddo.
You too, man.